Hey everybody. Sorry I'm 5 minutes late. I was actually sitting in another Zoom thinking that I had the right one, but I guess it was the wrong one. So I went to the Canvas page and this link was a different one than the one I had on my calendar. So sorry for being late. I thought just nobody was showing up. I was about to just call it off. All right, looks like a few people are still joining. We'll give them a sec here. Right. So sorry, there are a few of you that hopped on while I was saying that. Sorry, I was a couple minutes late. I was in another Zoom. That was just the link that was in my calendar was different than the one in Zoom first, or the Canvas page for some reason. So I was sitting there waiting for you, but I was in the wrong one. All right. So let's start out with any questions you guys have, and let's get going.
Do you think we could go over like the extra credit? Like I forgot what PowerPoint slide it was, but it was like the spinal nerve and with like all of them branching out.
Yep, absolutely.
Thank you.
Share my screen here. Make that a little bit clearer, I think. Okay. Is that somewhat clear? Can you guys see that? Yeah. All right, so yeah, so this here, there's multiple slides that kind of show this, but the bottom line thing to understand is right here that so we've got the dorsal side, which is the so well, let's go to the SAD Dave thing. That's kind of helpful, I think. So sensory equals afferent, right? So that just kind of tells you what that means, which you guys already know that. Equals dorsal, okay? But really the Dave part is the part that's the most helpful, I think, right? It's dorsal equals afferent, ventral equals efferent, okay? So that just helps you remember which side is which as far as where the different signals are coming from. And so what that really means is that anything that is in this root over here, in this dorsal root, is going to be sensory or afferent neurons, okay? Anything that's in this ventral root is going to be efferent, okay? And then we get out here, past this crossing point, okay? So we see this kind of like crossing point here. Anywhere past that crossing point, this is probably the best image of it. That's why I'm on this slide, because this you can actually see this the crossover in the different neurons, right? So if we follow this neuron right here, okay, so follow this one with me, you can see where my mouse is. You guys see that? Yeah, so if you follow that one, right, let's take it all the way. It goes here, and then it crosses over to the ventral side, right? Whereas this one right here, the top one, stayed on the dorsal side, right? So any afferent neurons that are in this ventral ramus had to come from the dorsal side, because all afferent neurons come from the dorsal side, right? And it's the same thing for the dorsal ramus containing any efferent neurons, right? So we can follow this one here. Again, this one stays on the ventral side, but then if we follow this one's going to cross over in this crossing point and get over to the dorsal side. Okay, so again, any efferent neurons that are in the dorsal ramus came from the ventral side. Any afferent neurons that are in the ventral ramus came from the dorsal side, just because that's where they come from. Okay, yeah. So when we go to all these questions here, this is really just something to try to get you to wrap your head around that idea. It's not, it's not necessarily crucial that you get every single one of these questions, but it's just as long as you got what I just explained there, then you have it. Okay. For some people, this is just kind of gets more confusing than it needs to be. But I created it just to give you a way to kind of work through those those kind of structural things that we have going on there. And the only reason the language is a little different, whether it's originate from or enter this diagram, is I was just trying to follow the direction of the signal, right? So when we talk about an afferent neuron, it's coming from the outside in, right? So that's where I'm talking about where do they enter this diagram, okay? But when it's an efferent neuron, it's coming from the inside and going out. So that's where I would say originate here and then go out. Okay, so that's kind of that piece. Yeah, what else would you like from that, Leah? Would you like me to talk more about that, or is that anybody else have further questions on that?
That was really helpful. Thank you.
Okay, sure.
I had a question about the principles of electricity, the resistance slide.
Mhm.
I'm not sure what page that's on for you.
This one. Yeah.
I think it's on page 9.
Yeah, I got it right here. Perfect. Okay.
Yeah, perfect. Okay, so it talks about the insulator and and it's has high electrical resistance. And I was confused by that because myelin, right? The myelin sheath, that's an insulator. So how does that have high electrical resistance? Like what electrical resistance is that like doing?
Yeah, so it's really just like a wire in your house. It's like an axon and a neuron is almost just like this, right? And that's why it's such a great analogy, is because the inside of the neuron or the inside of the wire is a good conductor, whereas the outside or the kind of covering of the wire or the neuron is a bad conductor or a good insulator, right? So in the case of the wire, it's copper on the inside, which is a really good conductor. In the case of the neuron, it's water, right? Water transmits or conducts electricity very well, right? And then as far as the wrapping, that's the good insulator, it's either rubber or plastic, as in the wire, but in the neuron, it's lipid, right? It's fat. So electricity just doesn't pass through lipids well, and so that's actually, so it's kind of weird in a way that a lipid or that a resistor or insulator helps us send a signal faster, right? Maybe that's kind of what you're getting at, Kai, is like, why does a resistor speed up a signal, right? Well, it's because let's see, probably the best slide to show it actually. What would be the best slide? Yeah, I have a little GIF on one of these. I think it might be back in section 4 that shows it the nicest. I think it's back on, I don't know, it must be a junction one. Does that one show it? No. I'm not gonna be able to find it, I don't think, right now. But pretty much what I was trying to find is pretty much what happens is if you have the axon like this, right? It's gonna pretty much be able to skip through the part that I really want an image to show you guys.
So,
hold on one sec. Go ahead, you can ask a question.
Sorry, yeah. So what like electricity is it resisting? Like, because you said the axon travels faster when it's covered with more myelin, right? So what is it resisting?
Yeah, so the electrical current is the ions passing through, okay? And so by changing the electrical current on the inside where the water is in the axon, that actually creates some current. It's going to pass through that water, okay? And I know that's a very like intangible concept because we're just really talking about ions passing across. So it's not like those ions go through the water from one place to the other. They don't move, but because they're electrically charged and because they move across the membrane, it changes the electrical properties of the water that's right there, right? So then the water gets kind of charged right there. It passes on to the water that's next to it. It's next to it. So as it's passing through that water, it's also trying to spread out, right? It's not just going straight down the axon. It's trying to spread out because that's just electricity is going to go in every direction that it can, right? And so as it tries to spread out, it hits up against that lipid, which is a resistor, and it stops right there. It doesn't let it exit that axon, right? So it keeps its strength by staying within that water and just goes down the axon instead of spreading out. And then it gets to a point, the little node, which is called the node of Ranvier, and I don't remember, I don't think you have to know that for this class, but it's a little node between the myelinated sections. And right there, that section is just packed with sodium and potassium channels again. So then we can recharge the current, and then it skips through that myelinated part again, gets recharged at the next one, it skips again. And that's really the way that it flows. So it's just trying to keep that current inside the axon instead of letting it spread out.
Gotcha. Okay, thank you.
Yeah, for sure. What
I had
else?
a question about a part in the study guide where it talks about the muscles we need to know for the elbow movement. Do we just need to go back to like last week?
Nope, that's another one that I forgot to take out. Yeah,
Oh, okay.
we-
So the
we
whole
don't have it.
section?
yeah, we don't have any muscles in this one, do we? Right, there's no muscles, right? Yeah, we talked about all the muscle ones last time. So yeah, there's no muscles at all as far as like naming of muscles and knowing their movements in this. Obviously, it's about muscles, but yeah, sorry about that, guys. That was another one. I had one of those in the last study guide too. Pull that up real quick here. Yeah, where is that? Oh, right here, spinal nerves. Yeah, yeah, that shouldn't be elbow and wrist movements. We've already dealt with that. Is it just that part right there? Yeah. Was there another one too? Is that it?
Thank you.
Yeah, sorry about that, guys. Yeah. You don't need to know that for this exam. Okay, what else?
I had a question about the absolute refractory period.
Yeah.
Is that the same as the hyperpolarization, or I guess just
Well, it's yeah, it's a good question. It's more so happening during repolarization. Let's go to this slide. Maybe in this slide. Yeah, so it's more so happening like in here. Right, so and if you're looking at this graph here, kind of, yeah, so here and here, definitely another action potential is not happening if you're up here. Okay, so that you're definitely stuck there. And then in this region, it's most likely not happening too. There's kind of a couple different variations of the refractory period, but we just really need to know that pretty much for this class, what you need to know is that before hyperpolarization occurs, we can't have another action potential. And that's because the sodium channel is stuck like this, right? So my mouse is, yeah, so up here, so when the sodium channel is stuck like this, then we're in the refractory period, okay? And so it's not until we get to hyperpolarization that we reset the sodium channel, right? And by resetting, we mean close the activation gate, this big one, and open the inactivation gate, so we get back to this point here. Well, I guess it would be B actually. In this one, you would get back to this point, right, where the activation gate's closed, inactivation gate's open, and that's really kind of one of the amazing parts about this when you think about what we talked about in the first day of class, like structure dictating function. The structure of this little protein that just happens to have two different gates like this. The only reason we have a refractory period is because of those two gates, right? And the only reason that that action potentials travel only one direction down axons is because of those two gates, right? So it's because of that structure, that unique structure having those two gates, we can have this inactivated state, which creates our refractory period, which helps our action potentials only go one direction, which is what makes sense to the brain. Maybe our brain would have came up with something else if it was getting signals both directions on axons, but it doesn't really make sense to the way that we understand it, right? So that's what the refractory period is all about.
Okay, awesome. Thank you.
Yeah.
I want to ask just like a clarification. Is B represent a hyperpolarization, sir? Or is it A? Because I got repolarization and hyperpolarization mixed up for what it represents. Any channel.
Good, yeah, good question. Yeah, so the in between these two, right, the ion flow is the same, right? Potassium is going out, okay, so that part's the same. The only place we can tell the difference is the state of the sodium gate, right? So the sodium gate is stuck in this position during repolarization before we get to hyperpolarization, right? And then when we move from here down to this hyperpolarized state, that electrical potential, or that membrane potential that's lower than resting, is actually what causes it structurally to change from this to this. All right, so B is more of the hyperpolarization one, whereas A is the repolarization one.
Thank you.
Yeah, and it's a good question, Michaela, to ask, but really hopefully understand it more than just matching it up, right? Because that's really what that's the questions will be more, won't be so much like match it up. It'll be like, you know, a question that gets at your understanding of this gate needing to be reset before we can have another action potential, and that's what causes the refractory period and all of that. So.
Sorry if this is like a repeated question, but so if B is the hyperpolarization and the sodium gate is opening, what is that exactly? I just don't understand what that exactly has to do with just like potassium moving out. Because in my notes I wrote down that the what is it? The hyperpolarization is just when it moves more negative towards resting potential. So how exactly does like the sodium tie in with all of that? That's just
like great. No, that's a great question. Great clarification. So to answer a question that comes before yours, which is what causes hyperpolarization of the membrane? That's potassium going out, right? So that's why we see potassium going out. So that's what causes it to go down further than resting membrane potential, right? But then the question of what does that have anything to do with the sodium channel resetting? That's because these proteins the shape of these proteins is dictated by the voltage across the membrane, right? So we have some proteins that change shape because we bind a phosphate to them, right? So like that myosin head, when we put a phosphate on it, it totally changes its shape, right? When we have that troponin inside a muscle, when we bind a calcium to it, it totally changes shape and pushes tropomyosin out of the way, right? So this is an example like that, but it's not changing shape because something binds to it. It's changing shape because of the electrical current around it, right? And that's the incredible part of this kind of biochemistry that we're talking about here is that I mean, it's just a string of amino acids, right? It's a string of hundreds of amino acids, and it just happens to be that they're sensitive to the electricity around them. And when that electricity changes a little bit, it totally changes the shape of these proteins to open a gate or close a gate, right? And so these are voltage sensitive proteins designed perfectly really to do exactly what we need them to do, right? And so that's the connection between hyperpolarization. So it's that hyperpolarized state, or that membrane potential of around -70 or less, that changes the shape of that sodium channel protein. Does that make sense, Julian?
Yeah, thank you. That helps a lot.
Good. What else?
Could you go over the clinical applications of extracellular potassium being too low?
Yeah, too low. Yeah, the too low part. So this part here, when it's too low, so this is all about changing diffusion gradients, right? So all of these are all about diffusion, or these two are definitely all about changing diffusion gradients, right? So we rely on having a strong diffusion gradient for potassium to move out of the neurons, to be able to repolarize and hyperpolarize, right? So what happens if all of a sudden, because we already didn't have too much sodium or too much potassium outside of the cell, but if we have almost no potassium outside of the cell, then that creates a stronger gradient for potassium to go out. Okay, so then if we go back to this slide here, instead of just getting down to -80 or whatever we get to, it's going to go even further than that, right? It might go down to -100 or -110, right? Because there's such a strong gradient for potassium to go out of the cell because we don't have any at all out there. So then we just we get further away from this dotted line, which is the action potential threshold, right? So it actually is like we inhibit action potentials just by messing with that diffusion gradient of potassium. Does that
Okay,
make sense?
perfect. Yeah, that makes sense. Thank you.
Yeah, sure. And it's the kind of opposite with this top one, right? This idea of having really high extracellular potassium is a more likely problem but more of a lethal problem to happen if all of a sudden we just dump in a bunch of potassium and we actually kind of balance out the amount of potassium on the outside that we have on the inside. Now we're just stuck up here, right? We can do the action potential, but we can't repolarize, we can't hyperpolarize, so we're just stuck in a depolarized state. So that means all of our sodium channels are going to look like this. So we're just stuck in the refractory period, right? And it doesn't matter what our brain tries to do. Our brain tries to send a signal down those neurons, but if all the sodium channels are stuck in the refractory period, we can't do anything, right? So we can't contract muscle, can't contract our heart, can't contract our diaphragm, and that's why it's a lethal problem. So you guys and gals that are nursing students, when you get labs on your patients, you're almost always going to see potassium levels on there, right? And this is why, because it's absolutely crucial to have healthy potassium levels for muscle and heart function.
So when you talk about extracellular potassium levels being too low and say that it causes paralysis, would that be flaccid paralysis or not necessarily?
Yeah. Where are you? I know I have the word flaccid on here somewhere, but where are you drawing on a distinction on that?
Yeah, you just say paralysis, but I'm just wondering if it's like taking larger depolarizations, would that just mean like it's not receiving those action potential?
Yeah, you're talking about for this one here? Or no, being too high?
Yes.
Yeah, so it just makes it harder to, yeah, so I mean, paralysis, it can cause paralysis, but it's probably more like weakness at first, and then if it's really extreme, it might turn towards paralysis. It's not the same type of paralysis as like a spinal cord injury where you just can't even, can't even contract anything. It's more like weakness, which could eventually turn to maybe paralysis just in certain muscles or things like that. But it's not the same as like losing all function to a limb or something like that. Is that
Got it. Okay.
what you're getting at?
Yeah, definitely.
Okay.
Thank you.
Yeah. What else?
Can we go over the graded versus the action potentials? Just a little confused about that section. I think it's towards the end. Or
Yeah, you're thinking from this stuff here, like this stuff or more? Yeah, so yeah, when we talk about graded potentials, we're really just talking about one key thing to think about is we're not dealing with those same sodium and potassium channels if we're talking about graded potentials. Okay, when we have those, I'm trying to think of a good slide to pull up to have a good picture of that. Let me just pull up the neuromuscular junction one because I know it kind of image that kind of shows what I want to show there. So when we look at the neuromuscular junction, see out here on the normal part of the membrane, we've got those normal voltage gated sodium and potassium channels. When we first looked at this shape didn't mean anything to you guys. Hopefully it does now that we're seeing that this channel looks very different than this one, right? So these are sodium and potassium channels in here that open up in response to acetylcholine. So these would be creating graded potentials. And then eventually that graded potential can spread far enough to activate these guys, and then we get our action potential. Okay, so that's one difference, is they're just caused by different types of channels. Okay, and so we'll go back to this one here. So, and then that's one thing that we there was new information on this slide in this section in the, this is the last set of notes we did on neurotransmitters and graded potentials. Chemical synapses, is this shows both of these are ways to cause depolarizing signals or excitatory signals. And so it could be sodium rushing in, which is exactly what we have in the neuromuscular junction, or it could be calcium rushing in. So that's new information, right? So that's a graded potential where it just goes up a little bit towards the action potential threshold, but doesn't necessarily cause an action potential, okay? Remember that these can be summed, which means we could have one bump here, and then before it gets all the way down, if we get another stimulus, then it'll build off of this one and move up. And as soon as it gets over that threshold, then it spreads to those voltage-gated sodium and potassium channels, and we get our action potential. Okay, so those two things are causes of this kind of signal, which is the depolarization, also known as EPSP. Okay, but then we can also have graded potentials in the other way, right? We can have graded potentials that move us away from an action potential, which would be like these, right? So those would all have to be things that would be leading us towards hyperpolarization or more negative membrane potential. And so that could be caused by the same old way that we've done it before during repolarization, hyperpolarization, which would be potassium leaving the cell. Or this new one is that we could do that also by chloride coming into the cell because it's negatively charged, right? So these can also be summed. We can put a bunch of these on top of each other and get the membrane potential way down here to -90. So then next time we get some graded potentials in this direction, they've got further to go to get up to the 55, to the -55, right? So it just takes, that's a way to kind of inhibit the activation signal, right? Because it just makes it longer to go from where they're at up to the action potential threshold. Does that make sense?
Yeah, that makes sense. Thank you.
Sure. What else? So yeah, one thing I would definitely make sure you understand I mean, this is always a rule of thumb in my classes, but wherever you see clinical applications, make sure you know those and understand the physiology behind those, okay? So got a lot of those here on the neurotransmitters, right? So there's a lot of information here on all these neurotransmitter slides. But definitely the most important part is the pathologies, as well as the way different drugs activate or deactivate these pathways. Okay, and being able to connect that back to this kind of physiology that we talked about with hyperpolarizing or depolarizing potentials is an important thing.
For the clinical application question about the ADHD and Adderall, from my understanding from lecture, for the second question asking about focus and stuff, I mainly got that dopamine dopamine controls like motivation of the frontal lobe. So is that pretty much like the central idea that we have to know, or is there like anything else?
Well, there's two different neurotransmitter pathways that it acts on, right? That's the important part. So both dopamine and norepinephrine are the important two pathways there, right? So and so if we look at these guys, get these out of the way for a second. Yeah, so norepinephrine in the prefrontal cortex is inhibitory. So if we go back to the slide that talked about norepinephrine, which was that? This right here, right? So it says in the CNS or PNS, right? And then we got yeah, it just says they can be excitatory or inhibitory, right? Depending on which part of the brain we're in. And in this case, we're talking about one of its inhibitory effects. So the prefrontal cortex, norepinephrine is being inhibitory then. So it blocks the reuptake of it, meaning that the norepinephrine that's released in the neurons there in the prefrontal cortex just stays out there for longer. So we're going to see more of its effects, okay? and its effects are to inhibit us from acting on certain things, right? So we're just getting so many stimuli all the time, right? We're getting so much stimuli coming in, and our brains have to be able to weed through that stimuli, all those different stimuli, and say, no, this is actually the one I want to focus on right now, right? So we have to be able to inhibit all of those stimuli that are coming in. And there's something about a brain with ADHD that doesn't work as well. The brain with ADHD doesn't have quite the same ability to inhibit all those other stimuli, and it's kind of trying to interpret all those different stimuli coming in instead. So we need some help there, right? So we need some help by this drug to block some of those stimuli, so you can just focus in, right? So that's the norepinephrine part. But then we also, ADHD patients also need some help being motivated to stay focused on that one thing that we're not filtering out, right? And that's where the excitatory part of dopamine comes in the frontal lobe. Okay, so norepinephrine helps us weed out all the unwanted and unneeded information, but then the dopamine helps us stay motivated and focus on that thing that we're trying to focus on. So it's really those two different mechanisms.
Okay, thank you.
Yeah. My wife helped me with that one because she's a psych NP and she used to work with ADHD patients all the time. It's actually a disorder that's one of the most treatable psychological disorders that there is. And most of them aren't quite as responsive to treatment as that one, but the meds are extremely successful in treating the condition. Obviously, you guys have heard of the abuse of the meds probably too, but if somebody really needs it, then it's super helpful.
I had a quick question on the chemical synapse. Do we need to know the neurotransmitter that's going from neuron to neuron there?
Like right here, you mean?
Yeah, more so the slide that your mouse is on right now.
Right.
Yeah. Yeah, you just said like CA plus releases the neurotransmitter from the synaptic vessels.
This is just kind of general. So this could be any of those neurotransmitters that we talk about at the bottom down here, right? So that's kind of the idea here is this is just a general chemical synapse. But then all of those different neurotransmitters at the end that are in the slides, you could fill in one of these, right? You could do that. So it could be any of these. And that's why when we get to this slide, there's 3 different ways that we deactivate the neurotransmitter. And then when we go to the neurotransmitter slides, inactivated by reuptake, right? That's just one of the options, okay? So they're all inactivated in different ways. Serotonin happens to be inactivated by reuptake, whereas other ones are inactivated, same thing, by reuptake. So reuptake is definitely the most common one, but there's other ones like acetylcholine. It's more, the breakdown of it is more important, like we talked about right here. So inactivation is by this enzyme instead, right? So this one up here is just a general one, but we can plug in any of those neurotransmitters to this set of slides, and then the only difference, another difference too, would be which ions is it that pass through here, right? And that differs depending on which type or which chemical synapse you're talking about too. You don't have to know which one would be which. You just have to know the options here that I give you for both depolarizing and hyperpolarizing.
Okay, awesome. Thank you.
Yeah. What else we got?
So action potential is caused by membrane permeability. Sorry, is that caused, or is that in the neuron, like at the axon hillox, or is that in like a cell?
I think you're referring to like this question here, like what causes action potential?
Yeah.
Yeah, so it's talking about in the neuron. Yeah, I mean, we're anywhere really. It's just talking about in any cell that has an action potential on it. That action potential is going to be caused by the membrane of that cell being opened or closed to certain ions, right? Namely sodium and potassium. Because as soon as we open up those sodium gates, whether it's a muscle cell or neuron, those are the two that we talked about that are electrically excitable. But smooth muscle is also that way. As soon as we open up the gates to sodium, then we get action potential, right? So that's what I mean by changes in membrane permeability cause an action potential. It's just that it's the opening of the membrane to those different ions is what causes the action potential. All right, what else, guys?
I have a question about the lab quiz tomorrow. So is it on like all of the upper limbs or just like the arms of it?
It's just over the arm part, so the forearm muscles, both anterior and posterior forearm muscles, and then the antebrachial muscles, which would be like the biceps, triceps.
Are your quizzes different than Dr. Mahoney's? Because hers said it was all of the upper extremities.
Dr. Mahoney's or Professor Mahoney's for my section or for her lab section?
For her lab section.
Yeah, they're all the same. Yeah. Okay. Where did you see that was like she just said that during the live?
No, she sent out an email I think yesterday or the day before.
Okay. Yeah. Yeah. No, it's supposed to just be over the forearm in the upper arm.
Okay.
And that's about like 12 or so muscles, right?
Yeah.
Okay. Yeah.
So she was saying that we'd have to study like the lab quiz from before and the new information. So we're just going to study the new parts that we've learned. Correct?
That's what I've been telling you guys the whole time.
Okay. Yeah, just to
I
make
hope
sure.
I'm not wrong.
Yeah, I was just.
If I'm wrong, like, then I'm not going to hold you guys accountable for it. But that's what I've been telling you guys the whole time. So, yeah, I'll double check right after we get off this. But either way, I'm not gonna go back on my word. I told you guys that if all those other ones are on there, but that's my fault, not yours. All right. Other questions on this stuff, guys? All right. Well, if we don't have any other questions, we can end it here. Any last questions? All right, guys. Well,
Go over.
go ahead. Sorry. Go ahead.
Yeah, just like the part with like receptor channels and like talking about the acetylcholine on the skeletal muscles. Could you just explain real quick on the just like a brief explaining like what the how you have your skeletal muscles. I think that's like mainly the one we're like focusing on for these action potentials, and then like on the smooth muscle, because just like to have a couple examples of it.
Not exactly sure what the question was. Are you talking about a specific day of class?
It's in on the membrane potentials.
Okay, membrane potentials.
Sorry. Yeah, just like just understanding the part where we were talking about the second message system, and I remember how you said we didn't need to know like the specifics, but just like getting a general understanding of what that encompasses.
Gotcha. I think that's think that's another one. That's it. Synapse and neurotransmitter. Oh yeah, this part here. These different, yeah, these different types of receptors and channels. Is what you're talking about? Okay.
Yeah. I
Yeah,
have like my
okay.
notes are they're not. I'm trying to read over them and they're just really not making sense to me.
Yeah, so pretty much this is just kind of a this is just kind of like a bit more background information on understanding, like, so we had just talked about graded potentials and action potentials, right? And then that kind of transitions to this idea of like, how do we open up these channels, right? Because these are all little channels. And the question is, there's multiple ways to get these things to open up, right? So the different options, because in order to have a graded potential or an action potential, we have to open these channels up, okay? And so that's really what this next few slides is getting at, is like, how do we get those channels to open up, okay? So there's one option for that is a ligand-gated ion channel. Okay, so a ligand is just a chemical that binds to a protein. It's a receptor. Okay, and there's really 3 different types in the body that we would use. One would be a neurotransmitter. This would be, again, when something released from a neuron, going through that space and then binding to the target tissue, and then that opens it up. So that's the example of acetylcholine. Okay, that's the example of actually all the neurotransmitters at the end of these notes. That's a neurotransmitter. It's a ligand that's going to bind to a receptor and open up a channel. And then it allows for that ion flow, and then we can get graded potentials, which will eventually lead to an action potential or inhibit an action potential. Okay. Also, hormones can do this, right? We're not really talking about that yet, but that's just another option of how we open up a channel, is through a hormone, or a neurohormone, which is similar. It's just that it's released from the neuron, goes through the blood, and then binds to one of these same types of channels that opens up the channel. Okay, so that's kind of just that's that type, the ligand-gated one, which is what all these neurotransmitters are, is they're different ligands to open up an ion channel. Does that part make sense?
Yeah.
Okay.
I think
Yeah,
it
go
was
ahead.
like it was just like the part where you had arrows, and I think I was just writing it too quickly, and
Talking about this one here?
yeah. Oh no, it was like the ligated, and that helps explain it better. That's where it was like tripping me up.
Correct. Yeah, so this is just the other option of how we open up channels. If it's still the idea that something binds to it, which could still be a neurotransmitter, but instead of directly opening up that same channel that it bound to, like it did here, right? So we see the neurotransmitter binds to it, it opens that one up that it bound to, right? So that's the direct one. But then the indirect one would be it binds to a different receptor, not the one that's going to be the ion channel, but a different one. And then it sets off this whole chain of events inside the cell, which one of the actions includes opening up a different ion channel. Okay, but we could also do things inside the nucleus, like activate genes or activate enzymes. Okay, so this one is kind of an indirect way to open up one of these channels, second messenger system, whereas the other one is a direct way to do it. So all of these neurotransmitters act in one of those 2 ways. They either open up the channel that they're bound to, or they set off the second messenger system and eventually open up a different protein that's the channel. And you don't have to know which neurotransmitter acts in which way, except for obviously the one we've been talking about the whole time, acetylcholine. That's a direct one.
Cool. Thank you.
Yeah, for sure. All right. So there any other questions?
Thank you, Dr. Sawyer, for your time. Appreciate it.
Yeah, in fact. All right, guys. Well, study hard, and we'll see you guys soon. Bye.
Thank you.
Thank you.
Bye, thank you.